Hey there, science fans! You're watching Hannah Ackerman, and today we're diving into the fascinating world of gene insertion. Picture this, a double helix, the blueprint of life, unzipping, rearranging, and changing the very essence of a living cell. That's gene insertion in action. Gene insertion is a cornerstone of modern biotechnology, enabling groundbreaking medical, research, and industry advances. But how does it work? Let's dive into the world of gene insertion in animal cells. So why go through all the trouble of inserting genes into animal cells? Imagine treating diseases, producing life-saving medications, or enhancing agriculture. Gene insertion allows us to produce proteins, study genes, and cure genetic diseases. For example, insulin production for diabetes patients. Inserting the insulin gene into bacteria revolutionized diabetes treatment. Gene insertion also holds potential for preventing diseases. By enhancing the immune system or correcting defects, we could eradicate inherited diseases. In agriculture, gene insertion enhances growth, disease resistance, and nutrition. The possibilities are endless, paving the way for a healthier, sustainable future. Let's talk about inserting genes into cells. Think of it like sending a message in a bottle. Sometimes you want the message received immediately, other times passed down through generations. Temporary gene expression lasts only for a short time. It's like sending a text message. Quick but not permanent. Stable gene expression integrates DNA into the genome, like carving a message into stone, permanent and passed down. Crucial for gene therapy, ensuring long-term presence of the gene. Now let's dive into the methods of gene insertion. First up, we have calcium phosphate precipitation, a classic technique that's been around for decades. Imagine trying to sneak a note past a security guard. You wouldn't just hand it over directly, right? You might disguise it somehow. That's what we do with DNA. This classic method uses calcium phosphate to neutralize the DNA's negative charge, allowing it to pass through the cell membrane. It's like coating our DNA note in a friendly disguise that the cell membrane security guard won't reject. Once inside, the DNA can then enter the nucleus and do its thing. This method is simple, cost-effective, and has been used extensively in research. However, it's not the most efficient method, as not all cells take up the DNA calcium phosphate complexes. Next, we have liposomes. Tiny bubbles made of lipids similar to the ones that make up our cell membranes. Now, remember how oil and water don't mix? Well, lipids and water don't mix either, which makes liposomes excellent vehicles for carrying DNA. Positively charged liposomes protect the DNA and allow it to fuse with the negatively charged cell membrane, delivering the DNA into the cell. Think of liposomes as tiny submarines carrying precious cargo, our DNA. These submarines can navigate the watery environment of our cells and deliver their cargo directly to their destination. This method is more efficient than calcium phosphate precipitation and protects the DNA from degradation. It's like giving our DNA note a special armored vehicle to ensure safe passage. However, it can be more expensive and requires careful preparation. Now get ready for some electrifying science. Electroporation is like opening a temporary door in the cell membrane using a jolt of electricity. Imagine you have a walled city and you need to get a message inside. Electroporation is like temporarily opening a gate in the city walls, allowing the messenger to enter. Electroporation uses an electric field to create temporary pores in the cell membrane, enabling DNA to enter. It's effective but can damage cells if not done carefully. This method is quick, efficient, and works for various cell types. However, it requires specialized equipment and careful optimization to avoid damaging the cells. Microinjection is a high-precision method that involves injecting DNA directly into the cell's nucleus using a fine glass pipette. Imagine you're a skilled surgeon and you need to deliver a tiny, delicate instrument to a precise location within the body. That's what microinjection is like. Microinjection involves injecting DNA directly into the cell's nucleus using a fine glass pipette. While precise, it requires skill and is slow, as each cell is treated individually. This method is highly precise and guarantees that the DNA reaches the nucleus. However, it's time-consuming, requires specialized equipment and expertise, and is only suitable for a limited number of cells at a time. Finally, we have viral transduction, a method that utilizes the natural ability of viruses to insert their genetic material into cells. Don't worry, we're not using harmful viruses here. Scientists have engineered viruses to be harmless couriers, delivering our desired genes without causing disease. This method uses engineered viruses to deliver DNA into cells. It's highly efficient for stable gene expression, but comes with challenges like immune responses and genomic unpredictability. Think of it like sending our DNA note with a special delivery service that knows exactly how to reach its destination. Viruses are masters at entering cells and integrating their genetic material, making them highly efficient vectors for gene delivery. This method is highly efficient, especially for stable gene expression, and can target specific cell types. However, it requires careful design and safety considerations, as viral vectors can sometimes trigger immune responses or integrate into unwanted locations in the genome. From treating diseases to enhancing agriculture, gene insertion has the potential to revolutionize our world. As we continue to unravel the complexities of genetics, we can unlock even more incredible applications for this groundbreaking technology. Understanding gene insertion opens the door to innovations in science and healthcare. Imagine a future where genetic diseases are a thing of the past, where we can grow crops that are resistant to climate change, and where personalized medicine is the norm. That's the power of gene insertion. If you found this video helpful, share and subscribe for more biotech content. Until next time, keep exploring the fascinating world of science.